So, hello, good evening, everyone. So, welcome to the next edition of the classes of physiology. So, today we will be discussing two topics. One is respiratory system, another one is renal system. So, the respiratory system is a big chapter. You know that it has a very important role to play when it comes to physiology. So, here basically you should know about the lung volumes and lung capacities, and then the regulation of respiration, dorsal group of neurons, ventral group of neurons, apneustic center, pneumotaxic centers. Those centers you should be knowing. The definitions of what is tidal volume, what is FRC, what is compliance, and chloride shift. Okay, Hamburger's phenomena, Bohr's phenomenon, all those things are very, very important. So let's start now. We will be discussing one by one, and as it goes towards the end of the session, we will be discussing the MCQs also. And coming to the renal system, you have to know the structure of complete nephron, cortex, medulla, the parts, the detailed cross section of cortex and medulla, and then the urine formation. What are the three stages of urine formation, the concentration of urine, and the applied aspect. So questions will be based on applied aspect. So let's go one by one, and towards the end of the session, I will be discussing MCQ. First, we will go with the concrete theory part. Okay, so let's start with the respiratory system. Right. So the respiratory system, so dear students, the normal respiratory rate is 12 to 16 times per minute. So inspiration followed by expiration. Inspiration, expiration. So one cycle of inspiration plus expiration is equal to one respiratory rate. So in an adult, 12 to 16 times it will be. And in the newborn babies, it will be much more higher. So this respiration, there are two parts of it. External respiration and internal respiration. External respiration is the respiration from outside, you take the breath inside. So it involves the exchange of respiratory gases, that is oxygen, carbon dioxide from outside you're taking, and it goes into your lungs and blood. That is external. So what is internal? That is the exchange of gases between the blood and the tissues. So that is the internal respiration. So at two different levels, your respiration is going on. So this is the basic terminology to begin with. Then again, phases of respiration. I told you there are two phases, inspiration and expiration. The terms, you know what is inspiration and what is expiration. This is the normal respiratory tract. It starts with your nose, oral cavity, nasopharynx, oropharynx, and then it goes into trachea, the windpipe, and then it opens into the lungs. You have got left lung, right lung, and below is your diaphragm. So this is your the basic respiratory tract, what you should be knowing. So one MCQ, what is asked is, what is respiratory unit? Okay, so the respiratory unit is at the terminal end of the respiratory system, terminal end. So the respiratory unit includes 5 things. The first one is respiratory bronchioles. So you are seeing here respiratory bronchioles. Then alveolar duct. Then alveolar sac. This entire thing like a bunch of grapes is called alveolar sac. Then antrum. The cross section of one alveoli is called as antrum. And the small what you are seeing is an alveoli. So at the alveoli level, exchange of gases is taking place. These are just like one small balloons. That's it. So alveoli level, exchange of gases is taking place. So what is respiratory unit? This all includes respiratory unit. Now, so we have just seen alveoli or alveolar sac. Okay, so this is the alveoli. Concentration will be on this now. So this alveolar cells are of two types, type 1, type 2. So type 1 are which forms a site of gaseous exchange between the alveolus and the blood. So from that balloon type alveolus, the exchange of gases is going into the blood. That is type 1 alveolar cells. And there are type 2 alveolar cells which secretes the alveolar fluid and surfactant. Very, very important. The type 2 cells are, one is doing the function of respiratory system. The type 2 is producing some magical fluid called surfactant. So what is the surfactant? We should know that. So please understand, surfactant is from type 2 alveolar cells. That we will again come back in short while. So the pulmonary arterial pressure. So you have lungs, the arterial pressure in the pulmonary. Systolic pressure is 25 mm of Hg. Diastolic pressure is 10 mm of Hg. And mean arterial pressure is systolic plus 1/3 of diastolic, which is equivalent to 15 mm of Hg. So this you have to remember this. Systolic 25, diastolic 10, mean arterial 15. And the pulmonary capillary pressure is 7 mm of Hg. So please learn this. These are the basic facts. It can be asked as a direct question also. Now, what are the muscles associated with respiratory system? So you have got inspiratory muscles, expiratory muscles. Two phases are there. So again, inspiratory muscle, which helps in inspiration, there are again two types. Primary inspiratory muscles and the accessory inspiratory muscles, or secondary you can call it. So the primary inspiratory muscle is a diaphragm. So in the photograph, the picture what I showed you one line like a diaphragm. So that is the primary inspiratory muscle. And then external intercostal muscle. These are very important. Diaphragm is supplied by phrenic nerve and external intercostal muscle is supplied by intercostal nerve. So please do not forget this. Next, coming to the accessory inspiratory muscle, these are the supplementary muscle which will help. They are not the major muscles which does the inspiration. The major muscle is diaphragm and external intercostal muscle. But other muscles which supports them, that is sternomastoid muscle, splenius muscles, and then anterior serratus muscle and elevators of scapulae and pectoralis. So these are the 4 muscles which are accessory. So this can be an MCQ. Which is the primary and what are the accessories? So 4 options will be given. You have to pick the correct answer. Then coming to the expiratory muscles, you have got internal intercostal muscle. So a point to be noted, external intercostal muscle is inspiratory muscle, whereas the internal intercostal muscle is expiratory muscle. So don't get confused. The name is same. One is internal, one is external. Then you have got accessory expiratory muscles and your abdominal muscles are there. So these muscles, a lot of MCQs are made from this. Do not leave this. Now coming to surfactant. So which we have just discussed that type 2 alveolar cells make surfactant. So what is the surfactant? What is the importance of surfactant? If surfactant is not properly produced by type 2 alveolar cells, what will happen? So first we'll know the definition. So pulmonary surfactant is a surface acting material. It is surface acting material and it decreases the surface tension of the alveolar membrane. So this is the main MCQ answer. So the options, if it is there, this is the answer. It decreases the surface tension of the alveolar membrane. And the surfactant, again, it is secreted by 2 types of cells. So generally we study type 2 cells. But in more detail, when you see 2 types of cells. The one which we have already discussed, that is type 2 alveolar epithelial cell. That is one cell. The second cells which produces surfactant is Clara cells. And these Clara cells are situated in the bronchioles. So do not get confused. Type 2 alveolar cells and the Clara cells are the ones which manufacture surfactant. And what is the purpose of surfactant? It decreases the surface tension of the alveolar membrane. So these are the 4 things you should remember. Now, what is surfactant? Okay, it decreases surface tension, but what is this? It is nothing but it is a lipoprotein complex. So when you know the composition, it is lipoprotein complex. It is formed of lipids, phospholipids, proteins, and ions. This composition is also asked. And 4 options will be given. The phospholipid dipalmitoyl phosphatidylcholine, DPPC. This is an MCQ. It's a major component of surfactant. This is a major component. So please learn that. So what does the surfactant do? So surfactant reduces the surface tension. So between the surface tension between the alveoli, and it prevents the lung collapse. Otherwise, lung is like a balloon. If surfactant is not there, it will fizzle out. The air will fizzle out and it will just collapse like a tire puncture. It will happen. Second function of surfactant is it stabilizes the alveoli to resist collapsing. So it will again same thing. It will not let it collapse. Third, hydrophilic proteins defend lungs by destroying bacteria and viruses. So in the composition of surfactant, you have got hydrophilic proteins that will kill the bacteria and viruses. So it's a natural mechanism to protect the lungs. So what does that function? Hydrophilic proteins. And it helps inflate the lung at birth and supports first breath. See, when the child is still in the mother's womb, to the placenta, oxygen is going. The first breath, the lung has to expand, right? That is just immediately after the birth. So this is a very crucial stage during the child delivery. The first breath has to be there, and that is for the very first time that it will open up. The lung will expand. So please understand, lung will expand during inspiration. Only the first breath of the child is done after the childbirth. Immediately, seconds. Then, if the surfactant is not there, what will happen? Respiratory distress syndrome. So not able to breathe properly. Respiratory distress syndrome. So please make a mark of this. This is an MCQ. Now, coming into the areas where MCQs are generated now. So respiratory pressures are there. Different pressures are there. Intrapleural pressure is there and intra-alveolar pressure. Intrapleural pressure, pleura means lung covering, outer covering, and alveolar means alveolar sac, what we have just discussed. So, the respiratory pressure, there are two types of pressures, intrapleural pressure and intrathoracic pressure. Second one is intra-alveolar pressure or intrapulmonary pressure. So, these two pressures, you should know that. Now, coming to intrapleural pressure. So, what is intrapleural pressure? It is a pressure between or in the pleural cavity. There is a pleural cavity between the visceral and the parietal layer. Lung has got two layers, parietal layer, visceral layers. Between the parietal and visceral layer, whatever is the pressure, that is intrapleural pressure. Two layers are there, okay? And this pressure is always negative. This is an MCQ. Intrapleural pressure is always negative, and the question comes like this: end of normal inspiration, it is -6. At normal expiration, it is -2. At forced inspiration, it is -30. So values keeps on changing. What keeps on changing? Intrapleural pressure. At inspiration, it is different. After normal expiration, it is different. And the third one, after forced inspiration, it is different. So please note these values. And this negative pressure will prevent the lung collapse. Then intra-alveolar pressure. It is the pressure inside the alveoli, the small balls, the grapes, what we call it. So, inspiration, at the end of inspiration, the pressure is -1. At the end of expiration, it is +1. So, there is a drastic change from -1 to +1. So, because of this intra-alveolar pressure, the balloon type, the sac, what you have seen, respiratory unit, it drives airflow in to suck the air in and out of the lungs. If you take it out of the lungs, it will flow. So, because of the intra-alveolar pressure, exchange of gases is taking place, enables gas exchange. So, which pressure? It is that intra-alveolar pressure. So, this is a very, very important pressure. Then the compliance, some definitions regarding that, right? The compliance. So, this compliance, so what is the definition of compliance? So, this compliance, you should know the definition, and then you should know what all things, the clinical anatomy of this. Just a second. Yes. Yes. So compliance. So what is compliance? What is compliance? Ability of the lung and thorax to expand. See, our heart does not expand much, but the lung expands. So the ability of the lung is called compliance. So the formula is change in lung volume per unit change in pressure. So it is change in volume by change in pressure. So the compliance is less, the stiffer the lung. So it's like a tight one. So the lungs will not expand. So this is very important. Stiffer lungs, compliance will be very less. So reasons lungs alone are more compliant are because no restriction from thoracic cage and no inertia, so easier expansion. But the lung is expanding nicely. Why? Because there is no restriction from the rib cage. Rib cage is not putting a pressure on that. It's a protective covering and enough space is there. Now in this room, I can spread my arms. I can spread my arms because the walls are not close to me. The rib cage is not close to me. Same way the walls of the room are not close to me. So I can expand my arms. So the lung can expand because the rib cage is not covering it. So this is the graph of the compliance in different conditions, emphysema, normal fibrosis. Now, the most important part of respiratory system, that is the lung volumes. So lung volumes and lung capacities. These are the two terms you should remember. Lung volumes, 4 are there. Lung capacities, 4 are there. The lung volumes, the mnemonic is TIER, tire. TIER. T for tidal volume. So what is tidal volume? Tidal volume is nothing. It's just like the sea wave. You can see the sea wave. Just a second. Now, PPT is not changing. Let me check again. Please let me know the slides are moving or not. Please put it in chat box. Yes, okay, fine. So, lung volumes. I was telling lung volumes are 4, lung capacities are 4. This 4 plus 4, 8 things will tell the total function of your lung. So, whenever you go for a total body checkup, your illness would have gone. So, for how to check all those, your lungs is properly working or not, what they will tell you is take a pipe and blow, and there is spirometer. So in the spirometer, you breathe in, you hold your breath, you exhale, 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 like that. Different maneuvers they will tell you to do in the spirometer tube. So based on that, the values will come. So lung volumes are 4, TIER, tire, that is a mnemonic. So T for tidal volume. Tidal, you are going to see beaches, right? You have seen the tides, high tide, low tide. So the wave will go up, the wave will come down. How much up it goes, that much down only it comes down. So that is normal breathe in, normal breathe out. How much you breathed in, that much only will come out. So that is around 500 ml, half liter. Please note this value, okay? 500 ml. The next is inspiratory reserve volume. Inspiratory reserve volume. So normal breathe in, normal breathe out. That is, you know that. Now inspiratory means taking in air. It's the reserve. Reserve means you're storing it. If you have extra money, you keep it in bank. So this is extra air. Extra air inspired forcefully after normal inspiration. So I breathe in. I am not exhaling. I am breathing in. And then with full force, I am breathing inside again. I breathe in normally and then exert the force. Maximum air, how much I can take in. That is inspiratory reserve volume, and that is up to 3.3 liters for 3,300 ml. Then expiratory reserve volume, breathe in, normal breathe out. And after breathe out, still I am able to exhale. Where is this air coming from? The air which was stored in my different parts of the body, that is all coming out and it is going out. So that is expiratory reserve volume. That is about 1000 ml. I hope I am clear. If you have any doubts, please put it in chat box. Then the residual volume is the amount of air. Even after forced expiration, I exhale everything out, still some air will be coming out. See, I am forcefully I am doing it. Still somehow some air will come out. That is residual volume. And that is around 1200 or 1.2 liters. So these values are very important, okay? 0.5, 3.3 in liters or ml. These values will come as MCQ bits. They will change the units also, so do not get confused. Now coming to lung volumes. Till now, lung volumes we studied. Now lung capacities we study. Lung capacities. So what are lung volumes? TIER. That you understood, right? 4 are there. Now lung capacity is either 1 plus 2, then 1 plus 2 plus 3, then 1 plus 2 plus 3 plus 4. What is 1, 2, 3, 4? TIER. T plus I, T plus I plus E, T plus I plus E plus R. When all 4 you mix up, that is your total lung capacity. So we'll read that now. So first one is inspiratory capacity. This is nothing but the maximum air inspired after normal expiration. After normal expiration. So I told you TIER. So first one, T plus I. T is tidal volume, I per inspiratory reserve volume. Tidal volume is 500 ml and inspiratory reserve volume is 3300 ml. So if you sum both of them, 3800 ml. So 1 plus 2 is inspiratory capacity. Vital capacity is 1 plus 2 plus 3, T plus I plus E. I hope you're understanding what I am telling. It's a shortcut method I am giving. So it is T plus I plus E and equivalent to 4800 ml. So what are the significance of vital capacity? It is decreased in respiratory diseases. So if someone is having a lung problem or not, how to know it? By doing all these lung volumes and lung capacities, the vital capacity parameters will be drastically low. That is what it indicates some problem is there. Then you have got functional residual capacity. This is 3 plus 4. First one was 1 plus 2. This is 1 plus 2 plus 3. Now 3 plus 4. That is ERV plus residual volume. It is equal to 2200 ml. And total lung capacity is 1 plus 2 plus 3 plus 4. That is 600,000 ml or 6 liters. So maximum 6 liters of air. Breathe in different parts of the body. You can store, you can exhale. That much only a human being can do. This is what about lung volumes and lung capacities. This is just for your references, nothing much. Force expiratory volume, FEV. So that is air expelled forcefully in a set time. So this is what the test they do. And what does it signify? Diagnosis obstructive versus restrictive disease, whether it is obstructive or restrictive. Something is blocking your respiratory system or it is restricting it. Second, respiratory minute volume will tell it is increased in exercise and decreases in disease. Maximum breathing capacity, it's reduced in respiratory diseases. And peak expiratory flow rate helps to distinguish between obstructive and restrictive diseases. So these things you should know it perfectly. This chart, you can take a screenshot. What is the significance of each one? Next is your ventilation. So ventilation is nothing but pulmonary ventilation. That is the respiratory minute volume. Volume of air moving into and out of the lungs. That's it. For example, I am sitting in a room. How much air is coming into the room and how much air is going out? Cross ventilation, if it is there, that is called as ventilation. So the normal value of pulmonary ventilation is 6 meters per minute. Air moves per minute during quiet breathing. Normally, when you are breathing, not running an exercise. Normally, when you are breathing, total amount of air goes into your lung and out. That is ventilation. Same way, alveolar ventilation, amount of air going inside and coming out of the alveoli, the balloons. That is 4.2 liter per minute. And then there is something called a dead space. That is that part of respiratory system where no exercise is taking place. It is there, it is not available for usage in the body. It is just there in the pipes and all. It is not used. Okay. So what is the means of alveolar ventilation to blood perfusion? And that is VA by Q, which is equivalent to 0.84. So please learn this. Perfusion ratio tells us, it indicates efficiency of gas. Gas exchange is taking place, but at what efficiency? It tells you about changes in the ventilation or blood flow to the lung. Is there change in blood flow? So the ratio increases if the ventilation is more, or without blood flow change, indicates whether your blood perfusion is properly or not. The next topic is oxygen hemoglobin dissociation curve. So this we have studied in blood chapter also. So oxygen hemoglobin dissociation curve is the relationship between partial pressure of oxygen and hemoglobin saturation. O2 plus Hb. How much it is binding, that is oxygen HBO curve. So it is relationship between partial pressure of O2 and hemoglobin saturation. So as you see in the graph you are seeing, it is an S shape. S. So it is a curve and shows hemoglobin has more liking, more affinity for oxygen. So this hemoglobin saturation between oxygen and hemoglobin, these are very important. Here I am talking about this part. These are all MCQs. If the partial pressure is 25 to 27 mm of Hg, 50% will be saturated. That is called P50. You see the graph. This is P50, where 50% of oxygen is saturated with hemoglobin. If the partial pressure is 40 mm of Hg, 75% are saturated. And the partial pressure of oxygen is 100 mm of Hg, 95% are saturated. So oxygen and hemoglobin, the saturation. Then there is a deviation towards the left or the right. The curve is sigmoid shape. So there are some factors which will push the curve towards the left and some which will push towards the right. That is an MCQ. Which are the factors which push towards the left and push towards the right? So right shift downward. It goes downward, it is the hemoglobin is releasing oxygen and more easily. So less affinity, the curve goes right shape. So it is caused by 5 factors. The 5 factors are here: decrease in oxygen, partial pressure, increase in carbon dioxide, that is called Bohr effect, the phenomena, increase in hydrogen ion, that is towards acidic condition it is becoming, or increase in temperature. So this is extremely important. Right shift and left shift are just opposite of each other. And learn this properly. Lot of MCQs you will find this left and right shift. What are the factors associated with it? Then a term called Bohr effect will be there. So Bohr effect is nothing. Carbon dioxide in tissue decreases the hemoglobin oxygen affinity. Hemoglobin is there. Oxygen, carbon dioxide. Because of the presence of carbon dioxide, hemoglobin affinity towards the oxygen is reduced, and that is called Bohr effect. That is called Bohr effect. Okay. Now, transport of carbon dioxide. So, how does carbon dioxide travels in your body. And in every cell of your body, when the metabolism is taking place, the end product is CO2. So it has to go to the lungs for removal and purification. The carbon dioxide produced in the body tissue is transported by the blood to the lungs for removal. And in four forms, it is transported. One is dissolved CO2, which is only 7%. As carbonic acid form, that is very negligible amount is transported. As bicarbonate, this is an MCQ. So as the bicarbonate, HCO3 minus 63% of the carbon dioxide, the waste carbon dioxide is sent out of the body in the form of bicarbonate, and that is the form in which it is transported. And then carbon amino compound 30%. Amino compound 30%. So these are the four forms how the carbon dioxide is transported. So carbon dioxide is transported in four forms. The most important form is bicarbonate. Now, coming to what is carbonic anhydrase role here? So this is nothing but it speeds up the reaction. It's a catalyst. It's an enzyme. So it speeds up the reaction 200 to 300 times compared to plasma. And this is nothing. The carbonic anhydrase has a role in your chloride shift or Hamburger phenomena. This is the Hamburger phenomena, what you always study. So this is nothing. Inside the RBC, there is H2O and CO2. So H2O plus CO2 will form H2CO3, bicarbonate. H2CO3 is highly unstable. So that will break down into H plus and HCO3 minus. And outside the RBC, you have got NaCl. NaCl will break down into Na plus and Cl minus. So HCO3 is minus and Cl is minus. HCO3 is inside the cell, Cl minus is outside the cell, RBC. So they exchange. What happened? HCO3 minus comes out because the minus negative anion has come out. Negative charge chloride goes inside. Exchange offer. So sodium and bicarbonate form sodium bicarbonate. Cl has gone inside and H plus was there. So H plus and Cl, they become HCl. Okay, so that is all about your chloride shift or Hamburger phenomenon. The chloride is shifting from the extracellular to inside RBC. So chloride shift or Hamburger phenomenon is the exchange of the chloride ions for a bicarbonate. Chloride will go, bicarbonate will come out. In RBC, that is an MCQ. Where does it happen? In RBC. Other parts, you can read like, and the reverse chloride shift is in the reverse order. Same thing what I narrated, in the reverse order is a reverse chloride shift. Same thing. Next term is Halden effect. The Halden effect is the effect by which combination of oxygen with hemoglobin displaces carbon dioxide from hemoglobin. That's it. So I am the hemoglobin. I am a very good friend with oxygen. But because of the presence of carbon dioxide, my association with my oxygen is not taking place. For example, you have two friends. Individually, you are friends with both of them. You are not, as a total, as a group, you three are not friends. One is your school friend, one is your college friend. Both are different friends. Because of the presence of the college friend, you are not getting enough time to spend with your school friend. Same example. The Halden effect is the effect by which combination of oxygen with hemoglobin displaces carbon dioxide from hemoglobin or college friend. That is what it is. So what is the significance of Halden effect? It is the for release of carbon dioxide from the blood. So the carbon dioxide has to be kicked out. So when the oxygen comes, hemoglobin will kick out carbon dioxide. And it is very important to release carbon dioxide from the blood into alveoli of the lungs and uptake of oxygen by the blood. So it's all the chemistry, make sure it goes out. So now some of the disturbances: apnea, temporary stoppage of breathing. So voluntarily, if you want to hold your breath, you can hold it to 40 to 60 seconds. But with practice, people do more with yoga and all. People can stop their breath for a longer period of time. Then this apnea, you can master it. And what you say, after hyperventilation, it is there. During the deglutition, when you are swallowing the food, you stop breathing for a few seconds. Then during vagal stimulation, adrenaline injection, and sleep apnea. So these are 6 conditions when apnea occurs naturally. So please learn this condition. This all can be an MCQ. Hyperventilation can happen because or during exercise or voluntary hypertension. Hyperventilation. In hypoventilation, suppression of respiratory centers, your dorsal group, ventral group of neurons, pneumotaxic, apneustic, and drugs affecting respiration, they can affect the hypoventilation. Next, coming to hypoxia. This is very important, this slide. In hypoxia, if you know this slide, it's more than enough. So this slide tells that there are 4 types of hypoxia. First one is hypoxic hypoxia. Second one is anemic. Third one is stagnant. And fourth one is histotoxic. So these are the features. So in which type of hypoxia, what is normal, what is reduced. So it's all self-explanatory. Nothing much to explain here. So please learn this chart. So many MCQs, almost like 15-20 MCQs from the same table can be made. Now coming to the applied aspect, there is something called as Cheyne-Stokes breathing. It is the periodic breathing characterized by rhythmic hyperapnea and apnea. Hyperapnea and apnea, both are there. Apnea means stoppage of breathing. Hyperapnea means you are breathing at a very high speed, high volume. So both are happening. So this is a periodic breathing, common type. Next is Biot breathing. Biot breathing is another form of periodic breathing characterized by period of apnea and hyperapnea. That was hyperapnea and apnea. Here it is the reverse. Apnea and hyperapnea, and there is no waxing and waning in the breathing. What is waxing and waning? Slowly increasing is waxing, slowly decreasing is waning. So this up and down in the breathing is not there here. In Biot breathing, it's sudden drop and then again sudden up like that. So there is no sequential up and no sequential down. It's drastic fall down and drastically up. Then carbon monoxide poisoning. The common source of carbon monoxide is the exhaust of gasoline engines, your gases, LPG gases. So various sources are there that you can read about. So what happens if there is carbon monoxide? If you inhale more. So if you inhale that, it displaces oxygen from the hemoglobin. So breathing difficulty comes. So oxygen transport and oxygen carrying capacity of the blood are decreased. Then you feel uneasy. So difficulty in breathing. So dear students, this were the most important things in the respiratory system. Please go learn. There will be some here and there some small things which I would not have covered today, but this is more than enough. And little bit about respiratory centers. We anyhow doing MCQs in that. I have included there, so I have not gone here. Read that part also. So coming to renal system. So what is renal system? This is the renal system, kidney, and you know the renal outer layer is the cortex, inner layer is the medulla. So this is the diagram. You should learn it. So kidney are 2 paired and they are situated where? In retroperitoneum, which is backside. And the weight is 150 grams. and there is the renal system. Like you have a music system, like that renal system. So this renal system are having some parts. Pairs of kidney, ureters, urinary bladder, and urethra. 4 parts. Functions of kidney, I did not explain. This you know that. So this is the most important. We have got outer cortex and then inner medulla, and there are spaces in between. That spaces what you are seeing is called renal sinus. The structure of nephron, right? So this the Bowman's capsule, and the cup is called glomerulus. Then you have got PCT, then thick descending segment, thin descending segment, hairpin bend, that is called loop of Henle. Then thin ascending segment, thick ascending segment, DCT, and then your convoluted duct or collecting duct, you can call it. So these things, and here in the Bowman's capsule, efferent arteriole will come and efferent arteriole will go. So blood will come and it will go back. But here, this is the blood vessel. Once the blood comes and it goes out, the liquid contained, the waste part of the blood is filtered over here. And that is how the urine is formed. The liquid comes out. Now, the nephrons, the structure, what is that? Functional unit of kidney is called nephron. So this nephron, where it is located in kidney? If it is in the outer cortex, it is called as cortical nephron. And if it is present in the juxtaglomerular apparatus, it is juxtamedullary nephrons. This is the parts of nephron which I already told you right now. Nephron is of renal, Malpighian corpuscles or tubular part. So as you see the diagram, this is the Malpighian corpuscle, and all this remaining part, this blue color one, is the tubular part. Then in the renal part, you've got glomerulus and Bowman's capsule, and in the tubular part, PCT, loop of Henle, DCT, thick and thin hairpin bend. So this is just the parts of the same diagram, what I have just told you. So structure of renal corpuscles, you have got glomerulus. So the cells you should know, inner cells, outer cells, and in between the space is there. Between the parietal and visceral layer, a space is there where the urine comes out. So you've got efferent arteriole which brings blood to the nephron. Efferent arteriole which pumps out the blood from the nephron. Glomerulus, it's a tuft of capillaries where filtration takes place. So these are all high yielding facts. What is the importance of each component? So glomerulus is the one where the filtration is taking place. Bowman's capsule is the first part of nephron where filtration takes place. PCT, selective reabsorption. So whichever is useful for the body, it will take it back. So that is PCT. Loop of Henle is important for self-gradient in the medulla. That is, the pressure is created over there. DCT, final selective reabsorption. These are all MCQs, okay? This part is significant for what? The collecting duct, it is for osmoregulation. The total length of the nephron is 45 to 65 centimeter. Huge. Here one scale is 30 centimeter. Imagine two big scale and even more than that. PCT is 15 mm and DCT is 5 mm. And these are the other measurements what you should know. Juxtaglomerular apparatus is just the same diagram. You see that there are two cells. So that is what the significance is studying in the Bowman's capsules and the glomerulus and the Bowman's capsule I'm talking about. So the structure of, or what is glomerulus apparatus? It is an apparatus, is a specialized organ situated near the glomerulus of each nephron. Just after a few minutes, we will be solving MCQs which are based on this. All things what we have discussed based on the MCQs, we are going to start very shortly. So structure of juxtaglomerular apparatus, three parts are there. Macula densa, extraglomerular mesangial cells, and your the juxtaglomerular cells. So same diagram what you have seen here. These are the glomerulus, mesangial cells. Between the efferent and efferent arteriole, some cells are present. Efferent and efferent arteriole cells are present. The cells are glomerular mesangial cells. So this cells are called granular cells also. This is what is important. So importance, right? So the cells are also called as lacy cells or agranular cells or Gormathi cells. You name it whatever. It's between the efferent arteriole, and also called as modified smooth cells, muscle cells. Various names are there. So basically, that is the importance what you should learn where it is located. Now, in the urine concentration, you have got three parts, right? First is filtration, then is the reabsorption, and then third is the selective reabsorption, the whole tubular part, tubular reabsorption. That each part, how the concentration of the urine changes. So this is all different MCQs had come at different times. A question will be given. The concentration of urine at this stage is how much? This is one chart. Take a screenshot. Learn this properly. You should be able to answer if your revision is proper. So basically, the urine pH is 4.5 to 6. It's slightly acidic. But the kidney prevents acidosis. Otherwise, if the acid is coming out in the form of urine, it will burn. Urine is acidic, but it will prevent acidosis by two methods. Urine. One is by reabsorption of bicarbonate ions. It will take back the bicarbonate ions and secretion of hydrogen ions. So it will take back, that is the reverse chloride shift will happen. And because of which the pH is not acidic, it comes almost towards neutral. These are the two reasons, very important MCQ. Then the facts about urine, the things you can read, the composition of urine, organic, inorganic content, the pH, osmolarity, color, order. These are the basic things. Then micturition reflex. So you should know what is this. It's the voluntarily passage of urine. So if you want to go bathroom, you voluntarily go. So there is a sensation goes towards the brain, and from there the impulses come. Relaxation of the sphincter muscles happen, and the voiding of urine happens. So that is called as micturition reflex. It's a reflex by which micturition occurs. It is stimulation of the stretch receptors, which is present on the walls of urinary bladder and urethra. So that starts with the MCQ now. Normal intrapleural pressure during the start of inspiration is dash mm of Hg. Please answer. If you can answer, it's very good. Don't see the explanation, and explanation is here, but don't see that if possible. Normal intrapleural pressure during the start of inspiration. So during the inspiration is different, and end of inspiration is different. That is, expiration is different. So at the start it is minus 2 mm of Hg. At the end it is again minus 2. At the start of inspiration is minus 2. At the end of expiration is minus 2. At the end of inspiration is minus 6. At the start of expiration is minus 6. It's different, right? So please learn this. So it is minus 2. Next question. Inspiration in respiratory function is monitored by. So your inspiration and expiration, two different phases. Inspiration is controlled or monitored by whom? So there is something called as inspiration, something called as expiration. Both are different. Control centers are different. They're asking the control centers. So you see the explanation. The medulla is the one which is responsible for controlling of respiration. There, two types of neurons are there. Dorsal respiratory group and ventral. Dorsal is responsible for sensory integration. So going information towards the brain only. And the function is solitary, like single. The ventral respiratory group is motor output. Location is thick nucleus ambiguous. So basically in the medulla, there is dorsal and ventral group of neurons. One is the stimulator, another one is the inhibitor. So this is nothing but the nucleus tractus solitarius, nucleus factors, sorry, various is the answer for your inspiration. Tidal volume in adult, this we have just discussed. So TIER, so the tidal volume is 500 ml. So this is the T plus I plus E, the lung capacities, and 3 plus 4, and 1 plus 2 plus 3 plus 4. TIER. 1 is T, 2 is I, 3 is E, 4 is R. So like this, learn the values and you can add all those things and derive at the correct answer. Same thing what I told you. Next question. A lecturer took the class to the students about respiratory function. Okay. He asked the students to analyze few cases. The analyzed respiratory function in 100 patients, they analyze students. And which of the following would best describe the restricted lung disease? Now, there are two types of lung disease, restricted and obstructive. Both are different. And for both, different parameters are checked. So they're asking for the restricted. Okay. So for the restricted, what we need to check is decrease in total lung capacity. Total lung capacity, it's gone. That is P plus I plus E plus R. 4 volume, that is equivalent to 6,000 ml per liter. Like that is what is not there. Decrease in total lung capacity is restrictive. It is a restrictive. The same thing. Total lung capacity in obstructive it is normal, but when restrictive it is decreasing. Residual volume in the obstructive it is increased, whereas in restrictive it is decreased. So please learn this. This is the clinical application of the values. So based on this only, when anyone goes for the total body checkup, the respiratory function test, they do it from this chart only. How it is, where it is, why it is, please go through this table and try to learn. Next MCQ. The alveolar ventilation in an individual with tidal volume 600. So the tidal volume is 600, dead space is 150, and the respiratory rate is 15 per minute. So what is the alveolar ventilation? So you should know the formula of alveolar ventilation. The formula of alveolar ventilation is tidal volume into respiratory rate. So here tidal volume is 600, respiratory rate is 50. That is pulmonary ventilation. But he's asking, the question is alveolar ventilation. Alveolar ventilation, the formula is equal to tidal volume minus dead space into respiratory rate. So tidal volume is 600, dead space is 150. So that is 600 minus 150 is 450. So 450 into 15, respiratory rate is 15. That is equivalent to 6.74. So this formulas you should learn. So please learn this formula. Next question. Oxygen dissociation curve shifts to right. So 5 factors are there. I told you these 5 factors. You have to learn that. So it's rising pH. So these are the factors. You can see this table. Shift to right, shift to left. All those things. Please learn. Do not waste time in this. Next question. Increase in partial pressure of carbon dioxide leading to release of oxygen of the tissues. Increase in PO2 of CO2, and because of which oxygen is released because of your school friend, your college friend you are kicking out. So that is your Bohr effect. You got bored. So Bohr effect. The presence of carbon dioxide decreases the affinity of hemoglobin for oxygen, and that is called as Bohr effect. And what is Haldane effect? Combination of more amount of oxygen with hemoglobin displaces carbon dioxide from hemoglobin. It's a reverse. Bohr effect and Haldane effect are opposite to each other. Okay, so please learn that. For Haldane effect, oxygen, and for Bohr effect, carbon dioxide. The type of hypoxia present in high altitude. So we have read one chart about that, right? So in high altitude, difficulty to breathe in is called hypoxic hypoxia. decrease in oxygen tension. So same thing, 4 types of hypoxia. Hypoxic hypoxia, PO2 of the arterial blood is reduced. In anemic, PO2 is normal, the pressure is normal, but the amount of hemoglobin is less. In stagnant, blood flow to the tissues is less. And histotoxic, oxygen is delivered to tissues in correct amount, but there are toxic agents, so the tissues cannot utilize the oxygen. So PYQs are here, flashed here, go through it. Next is surfactant is secreted by, so just now we have discussed that. This is type 2, so type 2 cells. The explanation I have already told. If surfactant is not there, respiratory distress syndrome will happen. Limitations of inspiration by vagal lung inflammation signal is called the Herring-Weir reflex. So it's a direct question. Herring-Weir reflex is lung inflation, inhibition of breathing. So lung inflation is stopped and inhibition of breathing happens. That is called as Herring-Weir reflex. Cosmos breathing respiration occurs in, this we have just discussed in our explanation also, decreasing pH of the blood. Decreasing pH of the blood or severe acidosis increases the rate and depth of respiration, and that is called as Cosmos breathing. End-tidal CO2 is decreased in all except. So end-tidal CO2, what it is a monitoring, it is a non-invasive technique which measures the partial pressure of maximum concentration of carbon dioxide. Normal values are 5 to 6% of carbon dioxide and equivalent to 35 to 45 mm of Hg. So please learn all these things. It will slowly you will go. Directly you jump into higher textbooks, nothing will happen. Step by step, slow by slow, you should increase your knowledge and then only it goes up. The renal blood flow to the kidney. For the renal blood flow, how much blood is coming to the kidney in 1 minute? So that is your 1260 ml per minute. In 1 minute, 1260 ml is coming. Normal adult value of urine output. So in a day, how much urine is output going out of the body? So 1.5 liters per day. That is the answer. Test for estimating kidney function test. That is called an inulin test. Okay. Go into the detail of inulin test. This is your inulin test. So it is a clearance test. Accurate determination of GFR is required over here. So what is inulin? It is a polysaccharide of fructose which is filtered by glomeruli but not reabsorbed, secreted or metabolically altered by the renal tubule. So that is the explanation. Inulin clearance is 120 ml per minute. Please learn this. Okay. Inulin clearance test. These are the PYQs. Go through that. So this class is anyhow there in YouTube. Please go back, take screenshots and go into the depth of this area. These are all areas, only those areas are focused which have come in the exam. Tubuloglomerular feedback. So this is from the urine formation. The tubuloglomerular feedback occurs in constant solute load on distal tubule. If the load is more, the feedback will go manufacture less. If the load is less, it will give an information manufacture more. That is the feedback. In which segment of nephron does tubular fluid has the highest osmolarity? So I told you the concentration of urine, one chart to remember. Take a screenshot, I told you that. That is Hende's fluid. These are all facts. Okay, directly you have to learn this. Most of the sodium is resorbed in where? Absorption of sodium is taking place in PCP. So like that, what happens in DCP, loop of Henle, other things, related questions also you should know. maximum reabsorption of filtered water occurs where? That is PCT. So draw a diagram of the nephron and each part, what is absorbed and what is out, you draw in the diagram and put it on your study chart on your wall. So repeatedly say you can answer this. In presence of excessive ADH, antidiuretic hormone, which part shows maximum hypertonic fluid? So that is your collecting TBO. Amount of glucose absorbed in PCT. So it's all high yielding facts. It's all there in your books. Straight away question, 100% is absorbed. So what are the substances which are absorbed in each part? PCT, DCT, convoluted tubule and loop of Henle. You should learn that which part what is absorbed. So you have got high threshold substance, that is glucose, amino acids and vitamins, low threshold substance, urea, uric acid and phosphate, and non-threshold substance which will just come out very easily is creatine. And the tubular maximum for glucose is 375 mg per minute. Right. So dear students, in the span of just 1 hour, we have quickly gone to 2 chapters, but there is a lot more to do than what is discussed. But almost 90% of the areas what comes in an exam is discussed. Some of the areas you may feel easy, some you may feel tough. So please go through this presentation again. Take out your textbooks and the areas which you are finding it tough, go back into the depth of it and learn it. Okay? And doubts also you can post in the group. Okay? So now go back and start reading. Thank you. Thank you, sir.